to continue our discussion, so we're gonna to Loeffler medium. So composition per liter. So beef serum 70 grams. Usually this is the source of protein. Egg and dried, so 7.5 grams. Also a source of protein and even fats. Heart muscle solids from infusion, so 0.72 grams. Glucose 0.71 grams. Peptic digest of animal tissues 0.71 grams, and sodium chloride dissolved 0.36 grams. So the pH is almost neutral or weak base at 25 degrees Celsius, so plus minus 0.2. So use for cultivation of Corynebacterium diphtheriae for demonstration of pigment productions, and the breakdown of proteins by Corynebacterium diphtheriae. So that is proteolysis process. The proteolysis, that is the breakdown of proteins. Okay, next. The Hoyle medium base, so composition per liter is agar 15 grams, beef extract 10 grams, peptone 10 grams, sodium chloride 5 grams, blood 50 ml, tellurite solutions 10 ml. So the pH is 7.8, it's almost basic, plus minus to a weak base, so that is 25 degrees at 25 degrees Celsius. Tellurite solutions composition is potassium tellurite 33.5 grams. So potassium tellurite is toxic for isolation and differentiation of Corynebacterium diphtheriae. For isolation and differentiation, so if you have the swab, so swab is coming from the sample or the patients. So you swab are cultured through tellurite agar or Loeffler pie slant. So we or from chocolate agar or blood agar. Then we examine for beta hemolytic streptococci for 24 hours, and then Loeffler pie slant. So stain with methylene blue. So sa methylene blue 2 to 8 hours. Then do the subculture to tellurite agar. To if they have the tellurite agar, so the black halo pigment is a indication that there is a presence of Corynebacterium diphtheriae. So you have to pick colonies from this subculture tellurite agar or tellurite agar, and then that culture is already indicated that there is a Clostridium diphtheria or diphtheriae. Then you do the inoculation, so suspected colonies onto Loeffler pie slant for the identifications, agar for in vitro toxigenicity. So meaning that in vitro, it is the study of toxigenicity of the diphtheria or microorganisms, or the toxigenicity test without using living tissues or tissues. So broth medium for in vivo toxigenicity. So this involved already tissues. For Loeffler or pie slant, so in almost 18 hours observation, so you do the biochemical testing and then smear with methylene blue to see if there is what we call a gram positive, that is almost a club shape, okay, club shape form of the Corynebacterium. So that is already the final confirmations based on the smeared stain with methylene blue, at the same time biochemical testing. So samples are coming from the suspected individuals or individuals suffering now symptoms of diphtheria. So diphtheria toxin. So structure, it is a heat label because toxin is a protein, a polypeptide. So of course polypeptide refers to protein that can be lethal, a dose of 0.1 microgram per kilograms. If disulfide bond are broken, the molecule can give two fragments. So now there is two fragments, A and B. So fragment B has no independent actions, but it is required for the transport of fragment A, because fragment B helps in the binding of the fragment B, I mean, helps the binding of fragment A in the receptors. Okay, so this is aided, I mean, for the transport. That is the functions of fragment B. Mechanism of actions: fragment A inhibits the polypeptide chain elongation by inactivating the elongation factor 2 or EF2. So later on we'll discuss that one. This factor is required for the translocation of polytidyl transfer RNA, that is transfer RNA to correct, from the acceptor to the donor site of eukaryotic ribosomes. Why? Because ribosomes is the center or the place for protein synthesis, or ribosomes functions for protein synthesis. So that is the main target of diphtheria toxin in the human cells, thus preventing protein synthesis, leading to the death of host cells, okay, or the human cells once it infect. So pathogenesis of diphtheria toxin. So we have here the fragment A and fragment B. So fragment A helps in the transport by binding to the receptor. So once inside fragment A, okay, so subunit B or fragment B, I mean, binding subunit help attachment to cell receptors. Okay? And ADP stands for adenosine diphosphate ribosylation of EF2 or elongation factor 2. And we have subunit A, active subunit cleaves nicotinamide from nicotinamide adenine dinucleotide and transfers remaining adenosine diphosphate ribose to elongation factor 2 ADP ribosylations, which inactivates EF2 and shunts or inhibits protein synthesis that will lead to cell death.